Hi all, test your knowledge with this microbiology quiz focused on microscopy principles, techniques and applications. In this quiz, you can answer questions about types of microscopes, staining techniques and their applications, magnification, resolution, contrast principles, etc. Like, share and subscribe for more microbiology quizzes and tutorials and turn on notifications to stay updated. Let's begin the questions. The first question is, unaided human eye has a resolution power of option A 0.2 millimeter, B 0.2 meter, C 0.2 nanometer or D 0.5 nanometer. The question is, unaided human eye has a resolution power of what? Is it 0.2 millimeter, 0.2 meter, 0.2 nanometer or 0.5 nanometer? So first, you should know about what is the resolution power. So resolution power is the ability of a lens to identify two objects as separate, two adjacent points as separate. So when it is not resolved, we can see a single object here. It is just resolved, that is, they are just getting separated. And when it is well resolved, we can see them as two separate objects. That is the ability of eye to see the objects as two separate points. That power is called as resolution power. And what is that power in unaided human eye? The answer is 0.2 millimeter. You can see when we observe through our unaided eye, we can see two objects as two separate points when they are placed 0.2 millimeter. And for light microscope, it is 0.2 micrometer, and in electron microscope, it is 0.2 nanometers. The next question is, resolution of a microscope improves by using what? Option A, oil, option B, stain, option C, lenses, or option D, condenser. The question is, resolution of a microscope improves by using which of these? Oil, stain, lenses, or condenser? And the answer is oil. By using oil, we can direct the light into the objective lens. So we'll get a resolution, better resolution. Without immersion oil, the light will be scattered. It will not be entering into the objective lens. So we will not get a better resolution. Only with immersion oil, we'll get better resolution. And what about stains? Stains are used for better visualization under the microscope for coloring the cells or cellular components. With unstained cells, it is difficult to visualize under the microscope. So stain is used for better visualization. And what about lenses? Of course, lenses are used for magnification. Eyepiece or ocular lens or objective lens, they are used for magnification to increase the size of the image. That is magnification. And what about condenser? Condenser is this part which focuses and concentrates the light coming through the stage or the light source so that the object will be correctly illuminated. So it is condenser. Next question is, phase contrast microscopy is useful for studying which of these? Microbial motility, shape of living cells, or bacterial components, or all of them? The answer is, it is used for all of them, because in phase contrast microscopy, the principle is a phase difference between the direct light and scattered light will produce a better visualization. That is why phase contrast microscopy is used. Next question is regarding gram staining. So in gram staining, which one is wrong? In gram staining, primary stain is crystal violet, B, mordant iodine, C, decolorizer sulfuric acid, D, counterstain safranin. Which of these is wrong? Which of these is not correct? The answer is decolorized sulfuric acid is not in gram staining. So in gram staining, the decolorizer is alcohol. Primary stain crystal violet, mordant is of course grams iodine, decolorizer alcohol, and the counter stain is safranin. So when we add crystal violet, both of these gram positive as well as gram negative organisms will be gaining the primary stain color, that is violet. Then when we use grams iodine as mordant, mordant is the substance which increases the affinity between the dye and the cell. So the stain will be more bound to the cell. So iodine is used for that. So all the organisms, gram negative as well as gram positive organisms, will be getting this violet color more strongly bound with the cell. And when we use decolorized alcohol, what they do is the gram negative cells will be decolorized. That is, the outermost lipid layer or the outer membrane, which is lipid rich, will be washing off with the lipid solvent alcohol. So only the gram positive cells will be retaining the color, and the gram negative cells, they will wash off the color of primary stain. And when we use counter stain safranin, which is in contrast in color with the crystal violet, what they do is they stain the gram negative cells. Gram negative cells take up the color of safranin and they will appear pink. And gram positive cells, they will be appearing violet itself, that is of crystal violet. This is the principle behind gram staining. So what are the uses of gram staining? A. Start empirical treatment. B. To detect yeast. C. Is for identification. Or D. Demonstrate motility. So what are the uses of gram staining? One of these option is not a use of gram staining. So start empirical treatment, detect yeast, identification, or demonstrate motility. Yes, of course, we cannot demonstrate motility with gram staining. For demonstration of motility, we use hanging drop preparation. That is, we take a drop on the slide and with a cover slip, we observe the motility of organisms. That is, demonstration of motility is not done by gram staining, it is done by hanging drop preparation. Thank you. Hope you learned something new today. Thank you.